Hello, everybody. Lori, thank you. Thank you. Thanks to the Molecular Frontiers Foundation and the Academy for the invitation to participate, and mostly just to feel like a student yesterday and most of today. I feel like I'm one of you. There's so much to learn from all these great talks. I'd like to start with maybe something a little philosophical, kind of big picture. You know, it's very hard to follow Lee because he's just reinvented all of medicine. So let's just kind of take a zoom way out and think about where we're all at. So this is our lovely, charming planet, teeming with diverse life. 7.5 billion souls on this planet. It's a remarkable place, and yet, there's a loneliness, isn't there? I mean, all this darkness, right? So we know about all of the other planetary bodies in our solar system, and now thanks to our planetary astronomer colleagues, the first of what will continue to be a deluge of extrasolar planets. But the fact is, as far as we know, we're alone. We're alone in the universe. Now, we may hope, even marshal statistical arguments, that that's not the case, but in terms of data, all 7.5 billion of us are alone on this planet. Don't mean to start with a downer, but that's just the fact. So how did we get here together? Well, this lovely planet of ours was formed out of the protostellar dust cloud of our solar system roughly 4.5 billion years ago, and it was not a habitable place. This is a phrase you hear now when people discover an extrasolar planet, whether it is potentially habitable. Our planet wasn't habitable for the first few hundred million years, but by about 4.2 billion years ago, the surface of the Earth had cooled to the point, accretion had completed to the point that it was possible to have a stable hydrosphere, liquid water, and it would be what from a distance would be called habitable. Now, then what happened, and Jack Szostak talked about this yesterday, then there was an era of prebiotic chemistry when things happened. Small molecules made more complex molecules. He gave you examples yesterday pointing more towards the molecule RNA. But in general, think of a whole range of chemistries happening on the planet or happening on materials that are delivered to the planet in an era of so-called prebiotic chemistry. And at some point, Darwinian evolution began, probably not of the same living system that we are all a part of, but of a simpler system based on RNA molecules. So this is what's often referred to as the RNA world. There's no direct evidence of this. When I say about 3.8, I really mean about 3.8. It could have been a bit earlier, it could have been a bit later. But by about 3.6 billion years ago, our life form, we're all the DNA and protein-based life form, all 7.5 billion of us, arose on this planet. And there's I think, good evidence in the physical and geological record that that was the case. So by 3.6 billion years ago or so, the RNA world was over. DNA protein-based life took over. And then the rest is phylogeny. It's just history of evolution. And here we are, right? How did we get here? Like this. And here we are. We're right next to corn. We're just down the street from paramecium. Okay, so that gives us some perspective on ourselves. So literally, from RNA to DNA and protein and on through phylogeny to us, we are the dust of the RNA world. It was almost 50 years ago that Francis Crick said this so beautifully and so correctly to this day that perhaps the first enzyme, the first catalyst, the first informational macromolecule of life was an RNA molecule with RNA replicase properties, an RNA that could copy RNA molecules, including copying itself. Dawkins in the selfish gene called this the replicator, in caps. The replicator. It had the extraordinary property of being able to create copies of itself. And what is the replicator that Francis and Richard Dawkins were talking about? It's the idea that RNA catalyzes the replication of RNA. So if we have parent strings of RNA that then are copied to make progeny strings of RNA, these are not just bits of information. These are molecules, and these molecules can fold into a shape, and by virtue of a particular shape, have a function. And if that shape enables the function, there it is, of catalyzing the copying of those strings, that's the replicator. That's the first enzyme that Francis Crick was talking about. And why is such a molecule so important? Because once you have an RNA enzyme that catalyzes the replication of RNA, you have Darwinian evolution. Right? You have the amplification of RNA subject to an occasional mutation, that encodes a function, and the better that function is at bringing about the copying, the more fit it is, and Darwinism is on. As Jack said yesterday, Jack Szostak said yesterday, the beginnings of Darwinian evolution is the beginnings of biology, right? So such a molecule gives you that beginning, and what this talk is about is having one of these molecules at our hand, right? We want to not literally reconstruct our ancestor, but show, in fact, that chemically it is possible for a replicator RNA molecule to exist. And as I say, because once you have such a molecule, Darwinism is on, right? So from among a population of variants, there's selection of the fit. Those become the parents that become amplified to make progeny, which can have new variation, and the Darwinian wheel turns and turns, and that's the phylogeny of life all the way up to paramecium, corn, and us. Now, as Francis Arnold talked about yesterday, there's a technology called Darwinian evolution, directed evolution, where we use Darwinian evolution as a technology to evolve functional molecules. Francis talked about the technology of evolving functional proteins. I'm going to talk about the technology of evolving functional RNAs, and in particular, evolving the replicator, evolving RNA enzymes that catalyze the replication of RNA, ultimately including their own replication. So what I'm going to do is I'm going to give you 3 examples of trying to build a replicator. Each in their own way can replicate RNA, an RNA enzyme that can replicate RNA. And I'll take you through each of those 3 examples. Each has their own strengths and weaknesses, and it kind of shows you where the field is right now. All right, so Jack Szostak talked about this yesterday. Fundamentally, what we're talking about, the chemistry that the RNA enzyme has to pull off, is a template strand of RNA binds a complementary primer, and then that primer is extended, copying the information on the template to a complementary copy. Now, as Jack explained yesterday, in prebiotic chemistry, one assumes that the monomers, the building blocks, were highly reactive because there were no enzymes. But in enzymology, in modern biology, one can use monomers that are something like the triphosphates, the nucleoside 5' triphosphates. N here just means any one of the 4 letters, A, U, G, or C, with a triphosphate group, a reactive triphosphate group at their 5' end. But reactive in the energetic sense, nearly inert in the kinetic sense, unless an enzyme, a replicator ultimately, comes along and puts these pieces together. Right? So that's templated extension of a primer, template-directed polymerization for a plus strand to make a minus strand. 2 strands come apart, then the minus makes a new plus. 1 makes 2, makes 4, makes 8, and off it goes. Now, if we do a kind of, I guess you would call it retrobiosynthetic analysis, right, and break down this process into something simpler, what we're really talking about is a templated joining reaction. So imagine if we just have 2 pieces of RNA that are bound on a complementary template that bring the hydroxyl group of one piece close to the triphosphate group of another, and we want an enzyme that brings about that joining, an RNA enzyme that brings about that joining. Now, I'm not going to talk about a lot of the details and the technique, but the fact is if one uses directed evolution, test tube evolution as a technology, one can develop RNA enzymes that bring about that reaction. So, for example, here's one of them. We call this the R3C ligase ribozyme. It was developed in my lab in 2001. And the bottom strand here is, we're calling that the enzyme strand. That's its actual sequence of RNA letters. And it binds 2 substrates, A and B, through Watson-Crick pairing to bind those 2 substrates, puts the hydroxyl next to the triphosphate, and makes that reaction go. Makes that reaction go millions of times faster than it would go spontaneously. And why we like this enzyme is it's small and it's very simple. It really is just kind of generically 2 regions of Watson-Crick pairing, a hanging stem, and a kind of critical set of nucleotides that brings about the function of the joining reaction. Now, the way in which the enzyme recognizes its substrates is through Watson-Crick pairing. The A substrate is bound through Watson-Crick pairing, the B substrate is bound through Watson-Crick pairing, and this part of the molecule does the work of the joining reaction. Well, now, what if the A substrate and the B substrate, when joined together, become another enzyme? Right? Because we can dial in different A substrates that match through Watson-Crick pairing and B substrates, but what if E, the enzyme, binds A and B, joins A and B together, and the product of that joining is another E? Well, then formally, we would have replication. So think of it this way. There's really 2 enzymes here, because there's Watson-Crick pairing between a parent enzyme and the building blocks that are going to make the progeny enzyme, right? So there's a Watson enzyme that's going to make a Crick enzyme, and then the Crick enzyme is going to make the Watson enzyme. So if we start with the Watson enzyme, it binds 2 pieces of Crick, right? And meanwhile, the Crick enzyme binds 2 pieces of Watson, and then Watson makes Crick, Crick makes Watson, and the 2 fall apart. 1 makes 2, makes 4, makes 8, and this could just go exponential. Now I'm going to show you actual data. This goes exponential. So this is time, and this is fold amplification, starting from 0.02 micromolar seeds of Watson and Crick, 5 micromolar of the building blocks of Watson and Crick, and it grows exponentially until the building blocks are consumed. And it could go forever, but it runs out of building blocks. Now, there are techniques that one can use to allow this reaction to go forever. One would be to set up what's called a flow reactor. So we would have a continuous influx of new building blocks, and the molecules would just keep replicating so long as that got supplied. Another way that's actually more convenient, and this is how we do it, is what's called a serial transfer experiment. So we take a test tube, and we put in it the 4 building blocks, the 2 pieces of Watson, the 2 pieces of Crick, a little magnesium salt, and then a little starting amount of the 2 enzymes, 1 or both of the enzymes, in liquid water. And we let them replicate, go exponential, until they run out of building blocks. Then we take a small aliquot out of that test tube and transfer it to a fresh test tube that has a fresh supply of the building blocks. And one can repeat that. That's called serial transfer. And so that mimics infinite space. Now, here's a real experiment where we did that. This is 50 successive transfers, 1 to 100 dilution. This is us just showing off here, but this is 10 to the 100th fold amplification in 37.5 hours. All right, so we learned yesterday from Dr. Gibson about what's the fastest doubling time for a bug, right? So Vmax is, what did he say? It's down to less than, how many minutes did he get to? 5 minutes, yeah. This one's 2.5 minutes, okay? But it doesn't have to make a whole cell. These are just molecules replicating like crazy. All right, well, there you go. So that's the replicator. That's what Crick was talking about. That's what Dawkins was talking about. And yet there's still a lot missing in this molecule because it doesn't have much capacity to evolve. The information that is being passed through the generations, through this exponential replication process, is only the information that is in this region between the Watson and Crick enzyme, the Crick and Watson enzyme. And that's only a half a dozen or so nucleotides in each of these 2 genetic loci, which can only encode modest traits. And so the bottom line is, although this molecule evolves in a Darwinian sense and can optimize, through Darwinian evolution, its function, it really only has the genetic capacity to optimize the function of replicating and can't do other things, like maybe ultimately invent DNA and protein, let alone process sugars and develop a membrane and harvest photosynthetic energy. So so that's the first story I wanted to tell you. I think we now need a kind of different perspective on things. And I show you the picture of this building. You're in this building now, but not this building. This is not the KVA. There's something wrong with this picture. Now, Bengt, you've been in this building more than 100 times, I'm sure, right? What's wrong with this picture? The crown is missing. Well, now let me give you a hint. It's wintertime, right? And look at the shadows. Does anyone see what's wrong with this? You're in this building right now. This is not the KVA. This is the mirror image of the KVA, right? The sun is in the south, not in the north. This is the KVA, okay? And I think that shows us something interesting about our own brains, how hard it is to even see that mirror symmetry. And in fact, when I was telling you about the replicating RNA molecule, you just assumed I was talking about D nucleotides, the same isomer that makes up the RNA in your own bodies. But I could just as easily have been talking about mirror image RNA, L nucleotides. I could make Watson and Crick and the building blocks of Watson and Crick out of L nucleotides, and by the rules of physics, of course, it would behave the same. And we've done that. And of course, it has to happen, right? So a D self-replicator and an L self-replicator behave the same. But we forget about the other side of the mirror. Now, what happens if we try to not just mimic one side of the mirror with the opposite-handedness of molecules, but actually try to play across the mirror? What if D molecules of RNA and L molecules of RNA are asked to try to talk to each other? Now, there's a lot of chemistry behind this I won't have time to explain, so I'm just going to state this as a fact, but you can look it up. It actually goes back to my own thesis work with Leslie Orgel. dRNA, of course, can form Watson-Crick pairs with itself. LRNA, by mere symmetry, has to do the same. But D and L-RNA cannot form contiguous Watson-Crick pairs with each other. All right? So D Watson and enantio Crick cannot shake hands. That's the bottom line, and vice versa. But what happens if we try to force the issue? What if we take a structured molecule of RNA, in this case the well-known HIV TAR element, and instead of making out a biological D isomer of ribose, or ribonucleotides, we make it out of the enantio L RNA. And then what if we go hunting using test tube evolution techniques for a dRNA molecule that can bind that LRNA molecule? Not by Watson-Crick, that's not allowed between D world and L world, but by some sort of tertiary interaction between D world and L world. The way we do that is develop what's called an aptamer. We immobilize this L molecule on a solid support through biotin. We make a large population of random 50-mer dRNAs flanked by primer binding sites. We select those that bind to the target, amplify the selected, repeat in a Darwinian sense, and develop so-called aptamers, only now these are cross-chiral aptamers, a D aptamer, trim it down, that binds LRNA and does so with an affinity of about 100 nanomolar. Okay, so this is D recognizing L, not by Watson-Crick, but by a cross-chiral handshake. And of course, if we flip the mirror, the behavior is the same, give or take a difference in synthetic purity of L nucleotides compared to D nucleotides. And the way in which these molecules talk to each other, as I say, is a three-dimensional handshake, not a Watson-Crick interaction. This is a protection assay, a kind of heat map showing what parts of these molecules touch each other in the complex compared to when they're individual molecules. The target is bound up in this loop region, and the part of the aptamer that binds it, highlighted in red, is this giant central ring. Okay, so that's how D can talk to L. Now, why did I tell you that story? Because let's come back to the replicator. What if we try to build a replicator, not in a homochiral world, but in a cross-chiral world? And why would we want to do that? Well, think about protein polymerases and the way they replicate nucleic acids. A protein can't play Watson-Crick with nucleic acids, it's a protein. So if you ask an LRNA to play as a polymerase with dRNA, it cannot do it through Watson-Crick interactions. It has to do it like a protein does, as a tertiary recognition of its target. All right, so we come back to this chemistry. Here's a template of RNA, two pieces of RNA that we're going to join together, but now we make it as L, not as D. Put a biotin to immobilize it on a solid support and link it to a big random population of D RNA molecules. And now we're looking for, among these random sequences using test tube evolution, particular D RNA molecules that will bring about the joining of LRNA molecules. And many generations of test tube evolution later comes such a motif. We then gave it an additional 30 nucleotides, more room to try to improve itself, more sequence room to develop more optimized function. We changed the format of the reaction. Now, instead of linking to the template, providing the template free and linking to one of the substrates. But again, if any sequence in here enables this reaction, the product becomes biotinylated, selected, amplified, and ultimately evolved to a more optimized enzyme, which we trim down. And now we have an 83 nucleotide D RNA enzyme that its function is to join together LRNA. Not through any kind of Watson-Crick interactions, but as a tertiary play. So let's just track the reaction here. We'll put a fluorescent label at the 5' end of one of the two substrates. And so if the reaction proceeds, that substrate will get larger by joining the two pieces together. We put a rhodamine label on the L substrate, and we see that over a matter of a few hours, the substrate then is joined to make the larger product. We can put a fluorescein label for D, and of course, the mirror flip is equivalent. So the D enzyme joins L pieces together, the L enzyme joins the D pieces together. You can even put both hands in the pot at the same time, and they're ships in the night. One does, the D enzyme works on L, the L enzyme works on D, and they make their products. Now that's just a joining reaction. We want to do many such joining reactions. So here's a case where using test tube evolution, we train this cross-chiral enzyme to polymerize trinucleotides. So here the building blocks are each 3 letters long. They each have a triphosphate at the 5' end, hydroxyl at the 3' end. And in order to get evolved in the test tube, the enzyme, which is of the opposite handedness than the substrates, has to string all these together of all different sequences to pick up the biotin, to get rewarded, to make progeny molecules and get evolved. And so that's how we further optimize this molecule. And now it's quite an efficient little cross-chiral assembly molecule for RNA, but of the opposite-handedness. Right? So here's a LRNA template, a long one, an LRNA primer, and lots of trinucleotides. And you can see that over time, these pieces, starting with the primer, get added successfully, and in a cross-chiral way, builds up an RNA product. Of course, ultimately, the thing you want it to build is itself. But remember, we're playing a cross-chiral game here. So what it has to build is the enantiomer of itself. Here's the enzyme. We can break the enzyme into, in this case, 11 different pieces, and then ask the D enzyme to join together the 11 pieces that will make the L enzyme. And that happens over time, starting with the first piece, all the successive additions, to make the full-length molecule, then purify that full-length molecule, and then it can do the same going back in the other direction of the mirror. All right? So this is another way to achieve replication of RNA, not in the conventional way, but this is where a right hand makes a left hand, and the left hand makes a right hand, and that keeps going. Now, at this point, though, this molecule, we're still optimizing it. It can add single nucleotides, but not as efficiently as short oligonucleotides. Trinucleotides are larger. So we're trying to train this to add single nucleotides much more efficiently. Why? Because that's the most information-dense way to do this. We want to have a template, remember, a primer, individual monomeric building blocks that will all be put together. So let me now turn to the third and final example of building towards the replicator molecule, but now in the more conventional sense. We're now back on one side of the mirror, and everything I'm going to show you from here to the end is D. We're in the regular KVA. The sun is where it should be in the sky. And we're going to develop a ribozyme that can very efficiently perform this reaction. And this last example is probably where the state of the art is in the field. This is the kind of top dog ribozyme right now. And it actually begins with another one of these ligases. Jack will recognize this molecule because way back when he was a teenager, Dave Bartel and Jack Shostak, out of random sequences developed through test tube evolution, this ligase ribozyme. Same chemistry, binds a piece of RNA, positions 3' hydroxyl next to triphosphate, joins the two together, nice catalytic rate. But this is a more complicated one than the one I showed you previously. And this is quite an interesting molecule because even as it stood from initial development, initial evolutionary development, it has the capability to add single NTPs to some extent. And with even just a bit of test tube evolution, was able to operate on a separate template primer and extend roughly a dozen or so successive NTPs on that template. Now, a scientist named Philip Holliger at the MRC Cambridge a few years ago published an advanced example of this molecule where what he did was he connected the ribozyme via a tether. Remember, we're all in D world here, so Watson-Crick is allowed. A little Watson-Crick binding domain, what's called a processivity tag, between the enzyme and the end of the template. So it hangs onto the template and doesn't let go. The primer binds to the template, and then the polymerase function extends that primer. And in this case, it can add tens, even more than 100 successive NTPs. So that's getting pretty fancy that you can add, 100 or more successive NTPs. Unfortunately, through all of its evolutionary history up to this point, this molecule had been trained on the same template sequence, a template sequence that was highly pyrimidine-rich and unstructured. And when given template sequences that are purine-rich or have any kind of structure, it doesn't work. But this is a job for evolution, for test tube evolution. And so published last fall, David Horning in my lab, after many years of effort on this molecule, developed what we call the 24-3 variant of this polymerase. It has 16 discovered mutations through test tube evolution, and it was trained to do the hard things, to copy not every sequence, but a broader and broader range of possible sequences. And it's now much faster. It's crossed the 1 a minute threshold for the rate at which it could add NTP, so it's a couple hundred times faster than its predecessor. But I think most importantly, it's now more general for template sequence. It can copy difficult sequences, purine-rich template sequences, templates that have stems and loops that otherwise would cause a polymerase to stop. This polymerase was evolved to get through that. It can't get through extremely tight structure, but it can read through structures as tight as this and through almost any sequence. Now, why is it so important to be able to read through almost any sequence? Well, if you can copy pyrimidine-rich templates to purine products, and you can purine-rich products, and if you can also then copy those purine-rich products back to pyrimidine templates, then you can go from Watson to Crick and Crick back to Watson, right? And the game is on. And so that's copying the copies, and that's, of course, what you need for replication. So this now optimized molecule can, to some extent, copy the copies. So this, everyone knows, I think, what PCR is, polymerase chain reaction. You would start with a template. You would bind a primer to the template. Only in this case, the template is RNA. The primer is RNA. The building blocks are RNA, the RNA NTPs. And the enzyme is RNA. So the RNA enzyme extends this primer on the template. And then that product binds a reciprocal primer, which the RNA enzyme extends and makes copies of the copies. We call this ribo-PCR, right? This is the PCR of the RNA world. Now, of course, there is a DNA-based human that is squirting things into the test tube, and there is a thermal cycler machine that we bought that was made by DNA-based humans. But once the reaction is running, it contains just RNA enzymes, RNA building blocks, a little RNA template starting material, RNA primers, and magnesium salt, and it just runs, just like PCR, only this is ribo-PCR. So it's not as efficient as good old PCR, the protein PCR, but hey, it's RNA. As Francis said, where's Francis? Yes, as Francis said, you know, let's face it, proteins are better. Yeah, but we had to start somewhere, okay? So here is a 1,000-fold amplification of RNA molecules catalyzed by RNA molecules. So that's a third way to do replication in a thermal cycling type environment, and Jack Shostak talked yesterday about imagining thermal cycles where in the cooler temperature, the primer is extended. In the warmer temperatures, they're separated. 1 makes 2, makes 4, makes 8, and so on. So that's ribo-PCR. But the fact is, this process is not yet good enough that we can do ribo-PCR of the enzyme that does ribo-PCR. If we did that, we would really be in good shape, right? Because then you would have a general purpose, self-sustained, evolving molecule. It can make pretty good sized segments, but you know, the longer the piece, the more structure, you know, there's a couple sequences that are more difficult. It can't make something as big as itself. So one game that we've just begun recently, and this is unpublished work, is what we call divide and conquer. So here's the same ribozyme that I just showed you that can do ribo-PCR, but we've now broken it into 4 fragments, right? So there's a break here, here, and here, so it's actually now in 4 pieces. But because of Watson-Crick interactions, those 4 pieces as separate molecules can come together, not by actually joining the bond at each of these junctions, but just by sticking together and then function as a ribozyme. So that means we don't have to make the entire ribozyme, we have to make each of the pieces of the ribozyme, and then once they're together in the same place at the same time, they'll come together and then be a functional ribozyme. And this is just showing the activity of the fully joined up ribozyme. So this is a primer being extended by adding NTPs to make a full-length product, time in minutes. If we break it into all 4 segments, it can do it. It definitely takes a hit because the break between 2 and 3, segment 2 and segment 3, hurts right now. We're now doing some engineering and some evolution to compensate or to improve the activity of the break between the 2 and 3 position. But that's another way to get there. If you can't beat them, you know, disjoin them and see if we can make these things in pieces. Meanwhile, the test tube evolution game continues. This is last year's model. This was published last September, the so-called 24-3 polymerase. The nomenclature here, 24 means the 24th generation of evolution in this particular study, and 3 refers to clone number 3. But here's this year's model, unpublished. We picked up another 13 mutations, going out another 14 generations, and it's a different sort of reference clone here. And this thing's starting to get really good. So here's what this one can do now. This is making tRNA. Okay, so this is yeast V tRNA. It's a complex sequence and obviously a complex and compact secondary and tertiary structure. This is a tough thing to copy. And the last year's model, the 24-3. So here's a primer, labeled primer being extended, and if you crank up the exposure, you can see it makes a little bit of tRNA. You can't see that, but if you have it on the screen and hit the contrast, you see a little bit of tRNA. But this year's model is doing much better. And now something that we consider kind of a watershed mark is, and now for the first time, the 38-6 variant, this year's model, can actually make grandma. Okay, it can, for the first time, synthesize its own ancestor. Because remember, this part of the story began with the Bartel-Shostak class 1 ligase ribozyme. The 24.3 just can't get there, but the newest version can actually take a primer, and in modest yield, just 1% or 2, but it can actually make the ligase that was the great-great-great-great-grandparent of itself. So that's kind of where things are now. We, of course, want to get to the point where it can make itself, the polymerase, not just the ligase, and maybe not the whole polymerase, but the fragments that can assemble to be the polymerase. So let me come back and end with the timeline again. Remember, we are here. We used to be here, we think. It is a hypothesis, though. Let's be careful, right? There's no direct proof that life on Earth came from RNA. There's a lot of indirect evidence to suggest that was the case, but there's no direct proof. It is a hypothesis. Now let's talk about, though, what had to happen right after that. To get from RNA life to our life form, two critical things had to happen, besides some amount of time, and we don't know exactly how much time that was. Two critical things had to happen. Protein had to be invented, DNA had to be invented. And I think we have a pretty good understanding of how protein synthesis was invented, because we can look in the ribosome and see that even today, the ribosome is an RNA machine, is a ribozyme that makes protein in an instructed way, instructed by RNA, adapted by tRNA molecules between amino acid and RNA information. So we can understand how an RNA world gave rise to protein synthesis component of a modern biology. But what about making DNA? All right, well, that's probably easy, right? If it can make RNA, it can make DNA. Okay, but just saying that is different than showing it. So I'm going to now show you, first time I've talked about this, a reverse transcriptase ribozyme. Reverse transcriptase means taking now an RNA template and making DNA from the RNA. So we have an RNA template, we have a DNA primer, and now the building blocks are NTPs, but they're DNA NTPs. And we now have, it's not as fast as our RNA polymerase, but hey, it's the first reverse transcriptase. So the RNA polymerase now is cooking along at 9 per minute NTP addition rate. But we now have a reverse transcriptase that's at a rate of 1 per minute. And of course, we're doing test tube evolution to improve that as well. It's not completely general with regard to sequence. Here's a variety of different template sequences that we've asked it to copy. Here's a primer that gets extended by adding deoxy-NTPs. This shows you where full length is. So some sequences it copies very readily to full length. Others it kind of chokes. This one sort of still has trouble with structure. It's harder to overcome this kind of structure than the RNA structure, at least for this ribozyme. But we're working on this one too. All right? So the goal is to keep improving the RNA polymerase ribozyme so that any one of the 3 versions I talked about today, so that they, in a more general sense, can copy, amplify exponentially RNA molecules, including themselves, or variants of themselves, to enable self-sustained Darwinian evolution. But also realizing we got to get from RNA to DNA, and so building an ever better DNA polymerase. And of course, what's the point of getting to DNA if you can't replicate DNA? So then we also eventually need a DNA-dependent DNA polymerase. And then, yeah, we got to invent protein synthesis, and then the rest is just history. Okay, so anyway, that's what I want to talk about today. As I said, the RNA world is a hypothesis, but the ability of RNA to act as a polymerase and the imagined replicator, I think, is not a hypothesis anymore. I hope I've convinced you that that's a chemical fact. And I just want to end by thanking people in my lab who did this work. As I said, I talked about 3 different approaches. The replicator molecule was first described by Tracy Lincoln and then optimized by Michael Robertson. This is our way of thinking about the cross-chiral polymerase. One hand draws the other. John Sapansky developed that, and Eric Mertfield optimized it. And then our ongoing saga of taking the class 1 polymerase to ever higher heights. David Hornings was most of the work I talked about. Katrina Chung did the ribozyme in pieces, and Biswajit Samanta developed the reverse transcriptase ribozyme. I, until the early part of this year, for many years, was at the Scripps Research Institute, but in January, it actually still says Scripps Research Institute on my badge, but I wrote Salk in, because in early January I went back home, so to speak, to the Salk Institute. You know, the motto there is where cures begin, and we hope someday it's the place where life begins. So thank you all for your attention.